There are 6 major elements essential to life: carbon, hydrogen, nitrogen, oxygen, sulfur, and phosphorus. But can an organism survive when one of these elements is replaced by one biologically similar? In a recent study, Felisa Wolf attempted to determine if bacteria can survive if phosphate is replaced with arsenate. Mono Lake in California has high arsenate concentrations and is home to the bacteria GFAJ1. Wolf hypothesized that arsenate can substitute phosphate in this species. To test this, she divided bacteria into 3 groups: a control group living in lake sediments that contained very little arsenate and phosphorus, a group in sediments with trace phosphorus and significant arsenate, and a group with no arsenate and substantial phosphorus.
The data she collected showed that less of the bacteria grown under conditions of high arsenate survived than those grown under other conditions, but those that did survive grew to a greater volume. These results are especially relevant to the study of DNA because phosphate is a key component of DNA's backbone. Wolf concluded that certain bacteria can supplement the phosphorus in its DNA structure with arsenic when it does not have access to sufficient amounts of phosphorus.
After the study was published, concerns arose in the scientific community as the presented results broke the long-established fundamental rules of life on Earth. In response to the article published, comments were made by different members of the scientific community to point out the errors shown in the study. One of the commenters, Rosemary J. Redfield, pointed out that various sources of contamination that would have made the conclusions of the study false. She pointed out the phosphate-starved cells presented actually were not completely lacking phosphate. The small, seemingly insignificant 3.1 microliters of phosphate in the growth medium would have been enough to recognize the cell growth that fueled the research article. Another major area of contamination included not washing the contaminated DNA and RNA pellets, not removing the gel slices from the chromosomal DNA fractions. Because of the errors in the procedure, Redfield commented on the unreliability of the published article.
Istvan Savai and Eor Sathmary also questioned the significance of such claims made by Wolf-Simon and her team. They cite the numerous inconsistencies in the report's data as cause for concerns about the accuracy of Wolf-Simon's data. Savai and Sathmary argue that these inconsistencies include, but are not limited to, large fluctuations in the reported concentrations and seemingly impossible error values. While there is not anything inherently wrong with large gaps in data values, Savai and Sathmary point out that such variation is not accounted for in Wolf-Simon's error calculations. They also highlight how the error margin of the percent arsenic by weight in arsenic-positive GFAJ1 samples is larger than the average percent arsenic. Not only is this seemingly impossible, as there can't be negative arsenic in a cell, but it calls into question the significance of the data relative to the null hypothesis, and therefore the validity of Wolf-Simon's claims.
Wolf notes about the lower-than-average uncertainties which may have been caused by the collection of samples when the cells were physiologically impaired. This impairment may have been due to the puncturing of cell membranes leading to a release of arsenic-containing compounds. One of the arguments posed to the experiment was that the analytical reproducibility had a 10% disparity that arose from each sample being of a separate biomass. Wolf accepts this argument as valid. She states that this argument is valid for arsenic levels, but the low level of variability in total phosphorus indicates that this error didn't affect phosphorus levels.
Wolf then goes on to defend her process for extracting DNA from Redfield's claims that DNA was not purified from the agarose gel, which may have led to impurities in the DNA that would skew results. Overall, Wolf's findings have been controversial to say the least. The recent calculations in quantum mechanics have indicated that the replacement of phosphorus with arsenic would not alter the double helix geometry or backbone of DNA. Wolf's experiment was not conclusive enough to nullify several years of scientific research. In order to account for the possibility of continued research, however, Wolf-Simon has made samples of GFAG1 available to the community upon request.